Hi, my dear students. Welcome back to Plus 2 Biology. Our chapter Human Reproduction. And today's topic is Spermatogenesis. So, in the last session, we had a discussion on the human reproductive system structure. Okay, structure of human reproductive system. And all of you have studied now. Okay, then you know which is the primary sex organ where sperms are produced. That means where the spermatogenesis takes place. Okay, spermatogenesis is the process of formation of sperms, and this process takes place within the testis of a human male. You know that. Okay, so where is the site of spermatogenesis? Yeah, it takes place, or the site of spermatogenesis is testis. Okay, and within the testis, exactly which is the location of spermatogenesis? You have studied in the detail and the testis cross-sectional view in the last class, and you know the testis consists of many compartments called testicular lobules. Okay, and within each lobule you have seen 1 or 2 or 3 seminiferous tubules. Don't you remember? So, within the seminiferous tubule only the spermatogenesis takes place. So, understand, within the scrotal sac there are testes. Okay, within each testis there are many testicular lobules. Okay, and within the testicular lobule there are seminiferous tubules. Okay, and the seminiferous tubules are the tubules within that the sperms are produced. So, the seminiferous tubule cross-sectional view, one portion enlarged is drawn on the board, and this diagram details also you have studied in the last session. Okay, so I think I can just recollect the things from you. Okay, got it? So, you have learned the cross-sectional view of seminiferous tubule in the last session. Okay, tell me what are the cells present inside the seminiferous tubule as the lining cells. The seminiferous tubules are internally lined by two types of cells. You can see here, these are one type of cell and this is the next type of cell. Tell me what are they named? What's the name of this cell? Seminiferous tubules, internally they are lined by male germ cells known as Spermatogonia. Is it not? What's the other name of male germ cells? They are called Spermatogonia. So keep in mind, seminiferous tubules are lined with one type of cell, Spermatogonia, and the other type of cell, which is a large cell known as Sertoli cell. Okay, so the seminiferous tubule is internally lined by two types of cells. One is the male germ cells, Spermatogonium. Male germ cell, Spermatogonium. Singular, Spermatogonium. The other one is Sertoli cell. Okay, Sertoli cell. And here you can just singular term, Spermatogonium. One only we have labeled here. Male germ cell, Spermatogonium. And from this name you have understood which type of cells are involved in sperm production directly. Among these two types of cells, male germ cell and the Sertoli cell, which is the mother cell of sperm? From which cell sperms are produced? Yeah, tell me. Yeah, sure. As the name suggests, Spermatogonium or male germ cell produces the sperms. Okay, then what's the function of the other cell? We have discussed in the last session. Sertoli cell functions what? Why these cells are there needed? These Sertoli cells function as nourishing cells. So understand, Sertoli cells provide nourishment to the developing sperms. Okay, whereas the sperms are produced from the male germ cells or Spermatogonia. Okay, that process only we are going to discuss in detail today. So, keep in mind, spermatogenesis takes place within the seminiferous tubules of testis. Okay, from Spermatogonia. Okay, the development starts from spermatogonia or the male germ cells. Okay, then just recollecting, tell me what are these cells? The cells seen outside the seminiferous tubule which are producing the male hormones. These cells are Leydig cells. Okay, Leydig cells secrete androgens or the male hormones. Which is the male hormone? Main androgen in male, testosterone. Okay, so Leydig cells secrete the hormones, which is very important for this process, spermatogenesis. The spermatogenesis is controlled by the hormones secreted by the Leydig cells. Okay, we will come to that. Anyway, I think this is clear, the introduction portion you have already learned. Let's come to the topic now, that is spermatogenesis. Okay, shall we proceed? Define spermatogenesis. Spermatogenesis is the process of formation of sperms from the sperm mother cells or from the spermatogonia. Got it? And where it takes place? Within the seminiferous tubules. Got it? Then just understand, spermatogenesis means genesis of sperms, formation of sperms. Okay, then let us discuss that process with the help of a flow chart. So here, first I am drawing a spermatogonium cell and I just represent it with a sphere. Okay, this sphere represents what? Spermatogonium. So this is one spermatogonium lining what? The periphery of the wall of internally lining the wall of seminiferous tubule. This is the wall of seminiferous tubule. This is the wall of seminiferous tubule and this is internally lined by the spermatogonium. Got it? So the spermatogonium, initially what happened to this is, it undergoes repeated mitotic division. The spermatogonium undergoes repeated mitotic division to form number of spermatogonia. Okay? And we can just categorize it into type A, type B spermatogonia. Some of these spermatogonia are retained for further mitotic division. They have to again increase the number. The spermatogonia has to be produced more and more because millions of sperms have to be produced. You know that. So some of these spermatogonia are retained for further production, and whereas some of these spermatogonia act as the primary spermatocyte. So understand, this spermatogonium by mitosis and differentiation. By mitosis, they are producing number of spermatogonia. Out of that, 50% are retained to continue the mitosis and they produce more and more spermatogonia. Okay, but if all of this spermatogonia are produced into sperms, from where the new sperms are produced? So, understand 50% of them are retained for mitosis and to produce more and more spermatogonia. Okay, to continue the process. Whereas, some of these 50% of these spermatogonia, they act as primary spermatocyte. Keep in mind, they act as primary spermatocyte. What's the ploidy of this cell? What is the ploidy of spermatogonium and what's the ploidy of primary spermatocyte? Are these cells haploid? Are these cells diploid? That's my question. Answer me. So, understand the primary spermatocyte and spermatogonia, both the cells are diploid. Okay? So, keep in mind, these cells, spermatogonium is producing primary spermatocyte by mitosis. So, this is diploid and this cell is also diploid. Got it? The spermatogonium is a diploid cell. Okay? It is producing spermatocyte, primary spermatocyte by mitosis. So, this is also a diploid cell. Got it? Then, this primary spermatocyte, it divides by, this is acting as the meiocyte. This primary spermatocyte divides by meiosis to produce two secondary spermatocytes. Understand, at first it completes meiosis 1. Meiosis has two divisions, you know. After completing meiosis 1, it produces two cells. We can call these cells as secondary spermatocytes. These are secondary spermatocytes. Give me what's the ploidy of this cell. Tell me, what is the ploidy of this cell? This cell is haploid. What's the reason? Here happened meiosis division, reductional division. So the ploidy is reduced to half. Got it? The first cell, Spermatogonium, divides by mitosis and produce large number of spermatogonia, and 50% of them act as a primary spermatocyte. They are also diploid and they start what? The first meiosis and produce secondary spermatocytes. They are haploid and these secondary spermatocytes complete meiosis. That means they undergo second meiosis. Okay. So when these cells undergo second meiosis, okay, what they produce? They are producing new cells named, you know the number. I mean, from one cell, two cells are produced. Okay, so they are producing the new cells named spermatids. What the name? They are spermatids. Spermatids are also haploid in number. Meiosis 2 division is exactly like mitosis. So it is same ploidy, N is retained. So things you remember, right? So understand, after meiosis 1, the cells produced are secondary spermatocytes, and after meiosis 2, the cells produced are spermatids. Okay, after meiosis 2, the cells produced are spermatids. From one primary spermatocyte, there are two secondary spermatocytes produced. You can see it here. Okay, and from the two secondary spermatocytes, there are 4 spermatids produced. So keep in mind, one primary spermatocyte produces 4 spermatids. So they mature to 4 sperms. That's what is the next step. Okay, what happened to these spermatids? Spermatids are not further dividing, they are just transformed structurally, I mean morphological change only. They are just modified or transformed to sperms. Okay, so understand, the spermatids develop to sperms. Okay, sperm. So each spermatid mature or they are transformed to sperms, we can say. The sperms we can draw like this. With a head, middle piece and tail. Okay. When you draw it beautifully and with all correct proportion. Okay. So one spermatid mature to one sperm. Okay. This is actually a morphogenesis process, and this process, this maturation process is called spermiogenesis. This maturation process, spermatids gets transformed to sperms by a process called spermiogenesis. These are sperms or spermatozoa. Okay, the spermatids mature to sperm by a process called spermiogenesis. Got it? So should I repeat it? It's a very important question, and mostly this question, I mean either spermatogenesis or oogenesis, is repeatedly asked for your board exam as a question. So you have to study this topic thoroughly with the help of this flowchart. You have to draw and practice this flowchart. Okay? It's very simple. So just understand, the starting cell is Spermatogonium. These cells are located towards the periphery of the seminiferous tubule. Okay? And this cell repeatedly undergo mitosis to produce more and more new cells. They are called spermatogonia. Type A, type B spermatogonia. Type A is retained for further mitotic division and producing the number more and more. Whereas type B is involved in this meiosis. So type A always undergo mitosis. This one undergo only meiosis. Okay, so understand, this type of sperm, 50%, suppose 50% of the spermatogonia, newly produced spermatogonia, they act as primary spermatocyte. Okay, and these are the meiocytes. These are the cells which undergo meiosis. So when they complete meiosis first, they produce secondary spermatocytes. Okay, and these secondary spermatocytes, when they complete meiosis 2, produce spermatids. Okay, since they are the products of meiosis, both the cells are haploid. The other cells, spermatogonia and spermatocyte, primary spermatocytes, are diploid. Okay, then what happens is the spermatids mature or transforms to sperms or spermatozoa, and this is what is the process? Spermiogenesis. The full process is spermatogenesis. The last step is spermiogenesis. Okay, in this series, keep in mind when the sperms are produced, these Sertoli cells give nutrition for developing sperms. That means the function of the Sertoli cells is to nourish the sperms. Okay, and initially when the sperms are produced, initially the sperm's head remain connected to Sertoli cells. So initially all the sperms, they remain their heads attached to Sertoli cells. You can see here. Okay, the sperm heads are embedded to Sertoli cells, and there is a process by which this is detached, and that is what is spermiation. Spermiation is a process of release of sperms from the seminiferous tubules, and they are free, not attached anywhere. Okay, so the last process, the name of the process is spermiation. Okay, what is spermiation? It's the process of release of sperms to the seminiferous tubules. From the Sertoli cells, they have to be detached and released to the seminiferous tubule, to the lumen, okay, to the central space of the seminiferous tubule. Now they are free for the transportation. Okay, so keep in mind the last process, from which it is detached to the lumen, is spermiation. So spermatogenesis, spermiogenesis, spermiation, these all terms you have to keep in mind.
Thank you.